Welcome back to Professor Yamuna Krishnan, now at the Department of Chemistry and Grossman Institute for Neuroscience, University of Chicago, for her lecture titled DNA and Nanotechnology Enters the World of Precision Medicine. This lecture is being co-hosted by ASET. For Sandhya Spaspa is the local host of the speaker. For some of you who don't know what ASET is, ASET at TIFR stands for Advances in Science, Engineering and Technology. ASET has taken its work in the form of Instrumentation and Electronics colloquia way back in 1983, and regular colloquia were held on every alternate Fridays. So somewhere down its journey, I changed its name to ASET to reflect its wider appeal and identity. So apart from TIFR colleagues, eminent speakers like what we have today from outside, not really outside, are also invited to speak in this forum. In fact, Yamuna spoke in the very same ASET colloquium forum while she was in NCBS way back in 2012, and to be precise, on May 4th, she talked about molecular DNA devices in living systems there. Now coming back again to InfoSys Prize, so on part of that I have a paragraph to read. Dr. Yamuna Krishnan is the winner of InfoSys Prize 2017 in Physical Sciences, and this is her award lecture. The InfoSys Prize is given to contemporary scientists and researchers below the age of 55 and consists of a gold medal, a citation by the jury, and a purse of 65 lakhs. The InfoSys Prize lecture series features talks by our laureates, like we have today, on their award-winning work and also by jurors on interesting work in their respective fields. These public talks are established by the InfoSys Science Foundation to spread awareness on the truly inspiring research and cutting-edge science being done by the scientists, and also to inspire young researchers and students make their passion for their profession. The InfoSys Foundation has created, in fact, a brief video presentation on Professor Krishnan on what inspires her by way of an introduction to her. So we'll play a 1-minute video before she can commence her work.
Me and my sister were always interested in sci-fi. So I remember at the age of 10 and 12, we were watching Star Trek. She said, I think one day there will be looking phones. This sort of vivid imagination was really important, and I think that put us on a path of science. My work involves making small structures using DNA like a filament to give you small devices that carry out specific functions. I really see a future where people with a large number of different diseases, neurodegenerative diseases, would be able to manage and control their own diseases on the back of this technology. I feel every day is exciting because we never know what we're going to learn.
Being podcasted live on ASET channel and as well as webcasted actually from Chai and Wise Facebook. So those of you who are outstation, of course, they can follow this talk on both these channels. So we now invite Professor Yamuna Krishnan to deliver her colloquia.
Thank you very much for that very kind introduction. I just realized that this is going to be a very general lecture, and so I just decided right now to change my talk to something that's more general, where I want to unveil to you recent findings from our lab that have happened over the last 6 months or so, where we can now use DNA nanotechnology to subtype neurodegenerative diseases. And to tell you what this is capable of, I have 2 neighbors, both of them fell sick more or less at the same time. One was my yoga master, and the other was a slightly older gentleman of about 65. They both had a battery of tests. They came back. The next 2 years was really interesting. My yoga master found out that he had diabetes, and he was in complete charge of his disease. He had his EpiPen, and he knew what was good for him, what was bad for him, and his disease was completely under control. On the other hand, my neighbor who was 65 years old, it took him another 3-4 years to figure out that he actually had Alzheimer's. But for a long time, we didn't know what was wrong with him at all. And even today, there's no cure for him. And so what I think we have developed is the basis of a technology that now allows us to take skin biopsies from patients with Alzheimer's or with neurodegenerative diseases, and we can actually see their responsivity to specific drugs. And this is the technology that I want to talk to you about. But it actually came about from very basic science, which had nothing, we did not envisage that this would be the end application. And so I want to lead you through how we got to this endpoint. And so my lab basically started by studying how organelle function impacts cell function. For those of you who haven't studied about organelles, they're subcompartments inside living cells. And just like organs of the body perform very specific functions for the body, organelles of cells perform very specific functions for cells. And so we try to understand how organelles function, and to do this, we use a new chemical imaging technology that's been developed by my lab. And it uses short sequences of DNA. These are about 20 to 30 base pairs. That's about 7 or 8 nanometers in length and 2 nanometers in width. So think of a cylinder with those dimensions. And these DNA duplexes act as fluorescent sensors for ions. And therefore, they can measure these ions very specifically inside these little compartments of cells. And what's unique about our approach is that because our probes image chemicals quantitatively, that is, it tells you the concentration of the chemical, not just this chemical is high, it's low, somewhat high, somewhat low, it's not that, it's a number. And so because of that, it gives us the ability to interrogate organelle function inside living cells and living organisms in very new and unusual ways. And so our philosophy is that because we can now see chemicals that we've always wanted to see in places of the cell we couldn't access before, we must be able to uncover and understand some new aspect of cell function. And so using this technology, what I'm going to talk to you about is how we can now map the metabolic status of cells derived from human patients. You see, the cell itself is like a chemical reactor, right? And so while it's reacting, it's producing all these byproducts. So just in the same way as if you have a car that's running and you keep sensors near the exhaust pipe, which is measuring chemicals, you can tell the normal function of the car or not. In the same way, as your cell is functioning, if you put sensors inside specific compartments of the cell that measure these chemicals that are being produced during cell function, you can get an idea of the normalcy of that cell. And so this technology now allows us to subtype neurodegenerative diseases. And so there are 4 different editorials last month in some magazines that you read that have discussed now how this technology has brought DNA nanotech into the world of precision medicine, and that's what I want to talk about for the first time today to this audience. And so I want to thank the Infosys Foundation for the opportunity to actually come back home and talk about this where it all started. And I also want to thank Sandhya for being an awesome host, and all of you for coming to this talk. It's great to see many of you all again. And so to get back to our talk, I wanted to say that just to remind you, we use fluorescent probes to study organelles, and one of the organelles that we're going to talk about today is an organelle called the lysosome, which is you can consider it to be the stomach of the cell. It degrades material, and it does so and gives the nutrients of incoming cargo to the cell for the cell to grow. And so as a chemist, when I first joined NCBS and I looked around and saw these biologists around me, and I would see their talks, and I used to wonder, why does the cell have so many compartments, right? Why so many? And so if you look at it, each organelle is actually a small reaction vessel. And as you know, as chemists, one reaction condition doesn't do all reactions. You need different reaction conditions to promote different reactions. And that's exactly what's happening inside the cell as well. So each of these compartments is carrying out a different reaction. For example, in the endoplasmic reticulum, amino acids are being stitched up into proteins. In the lysosome, you have proteins being broken down into amino acids. You have nucleic acids that are coming in that are being broken down into their monomers. In the Golgi, you have proteins that are being glycosylated. In the peroxisome, you have very, very long lipid chains being peroxidized into smaller lipid chains. So you can see that the chemistry that's happening inside each of these organelles is completely different. And so over evolutionary timescales, the milieu inside these compartments has been optimized to bring about that chemistry. And so what we thought was, if we can measure the chemical environment inside these organelles, that might give us a clue as to understand the biochemistry that's occurring inside the organelle and how that biochemistry drives the function of that organelle, and thereby how the organelle function drives cell function drives tissue function then drives organism behavior. And so I've wanted always to connect ions all the way to behavior. And so I also described to you how the ionic milieu inside is actually a steady state of that organelle's function, right? So it's basically, you can look at the metabolic status of that organelle inside a live cell. And so the organelle I'm going to talk about today is going to be the lysosome. And so how do you then measure a chemical inside an organelle, right? And so how we do this is the following. We take a simple DNA duplex, right? Watson-Crick base pair duplex. And on this duplex, you can have one dye that is sensitive to the ion that you wish to measure. So this dye can either turn off or it can turn on, right? So it can sense ions in two ways. It can either be a turn off sensor or a turn on sensor, where green fluorescence will go off if it's sensing, it's a turn off sensor, and if it's a turn on sensor, the green fluorescence will turn on, okay? And now because Watson and Crick base pairing is inherently 1:1, you can take another strand that's carrying a reference dye which glows red, whose fluorescence doesn't change at all. And so if you take make a complex between these two, you'll immediately see that the ratio between this red to green, so let me call this a sensing module and a normalizing module or a reference module, then the ratio between these two allows you to actually measure the concentration of your ion that's going to be in the milieu. So if you have no ion that's present in the milieu, the system will glow green, and then you have a gradual increase in the component or decrease in the component of green as a function of your ion concentration, where you can go from green, lime green, lemon yellow, yellow, light orange, dark orange, red. And so your color, your ratio between red to green is a direct measure of your ion concentration. So now you want to be able to measure this inside your organelle, inside your lysosome. So for this, what you need to do is to be able to position this measuring module inside this lysosome. And so for that, we developed a method to target these DNA nanodevices and position them inside the lumen, inside the hole or the space where the biochemistry is happening, inside the reaction compartment. And so how you do this is you take this targeting module and you you design it such that it can attach onto a particular protein that is going to be a carrier protein or a shuttle protein. And this shuttle protein is going to carry this DNA device from outside the cell culture milieu, outside the bath that you are soaking your cells in, and position your DNA device. It's going to grab onto your DNA device and position it inside your target organelle. How does it do this? So let's say, if I were to summarize whatever we've done, the overall concept is the following. If I want to target this device into a target organelle, I identify on that organelle a protein called a shuttle protein. And a shuttle protein will have actually two characteristics. One characteristic is it will have a very high abundance of shuttle protein on your target organelle of interest, and a very low abundance of shuttle protein on the cell membrane. So you might want to think of this as, if people want to come to TIFR, you have a lot of buses, shuttle buses of TIFR stationed at TIFR, and maybe one bus in Santa Cruz Airport, right? And then you have an active exchange between these two populations, where at steady state you always have one bus, empty bus, at Santa Cruz Airport, and all the remaining buses at TIFR. Then people who get down at the Santa Cruz Airport will who have a TIFR ID card can get onto that bus and come here. So if you wait a little while, whole of TIFR will become enriched with people who have TIFR identity cards. So it's as simple as that. So what you do here is you make sure that you have a measuring module that attaches to your shuttle protein through molecular recognition. And if you wait a little while, every single guy goes out, grabs a DNA sensor, brings it back inside, and now your target organelle inside a live cell will glow brightly, and it's like a fancy cuvette, fluorescence cuvette. So what is this? Yes.
Sorry, quickly.
Yes.
Can you go back to the slide?
Yes. Yes.
So there's just one quick question.
Yes,
on the
sure.
Sensing module.
Yes.
When you say the concentration gradients are understood,
Yes.
Aren't you limited by the optical sort of line bits that you get? Because your concentration resolution needs to be as precise as you want to be, of course, in a cell.
Yes,
If
yes,
you want
yes.
nanomolar changes to be detected versus micromolar, you can
Yes.
give away some of the resolution. But
Yes.
but how do you do that for different analytes?
So what you can do is, that's the next slide.
Okay.
So chemists, right, have, thank you for the question, which is, I was asked to repeat the question, which is, how do you decide what is the resolution to which you wish to measure your analyte of interest? So chemists have made a range of chemistries, a range of ions and small molecules that can detect analytes to very specific affinities and sensitivities. And so if you develop a platform where you can take any chemistry you wish and stick it on your DNA, once you know whether you want to measure in nanomolar or whether you want to measure in micromolar, you choose your chemistry. It's all there, it's on the shelf. And it's been on the shelf for the last 20 or 30 years. And that is something that I will try and describe to you a little later in the lecture. And so you have all these wonderful chemistries. The problem is you take these chemistries, these small molecules, and you add them to a cell. And these molecules basically paint the entire cell, right? So you lose what is most important, which is spatial resolution. I want to know what's going on in the lysosome, but I can't see the lysosome specifically, right? I see the whole cell. That is a problem. The second, biologists have been using fluorescent proteins, and this gives you exquisite spatial resolution. I know exactly what organelle I'm looking at. The problem is, they don't have the requisite chemical sensitivity. So if I want to measure zinc, well, there's no a fluorescent protein that is a zinc sensor. So this is a problem. I have spatial resolution, but no chemical sensing. And so what we try to do with DNA devices is to get the best of both worlds. By taking this small molecule chemistry and gluing it onto a DNA device, you can localize this DNA device in a specific part of an organelle And thereby you get here the spatial resolution that is accessible to a biological macromolecule. This is also a biological macromolecule. So it gives you stable spatial localization, but it also gives you not just chemistry, but because you can also attach a reference module, you can get a number out of it. And so the question immediately is, how does the targeting work, right? So I'll give you a range of methods by which we can target. So if your shuttle protein happens to be, this is just for the aficionados in the audience. If your shuttle protein happens to be an endocytic receptor, every endocytic receptor has a ligand. Simply glue your ligand onto your DNA device and it will engage your receptor and get localized inside your organelle. Immune cells, which is basically cells in your blood, express a particular receptor called a scavenger receptor that takes a very specific path: early endosome, late endosome, lysosome. This is called the endolysosomal pathway, and it's conserved in all immune cells. And these scavenger receptors bind anything anionic. DNA is anionic. So DNA is a wonderful substrate. And so this is a simple way by which you can add a DNA device. It will bind an immune cell or a skin fibroblast, which also has these receptors, and just localizes it into your lysosome. But there are many organelles and many shuttle proteins that don't have ligands because they are not endocytic receptors at all. And for those, we developed proteins through a phage display method that can bind a very specific sequence of DNA. And so now you have an artificial receptor for DNA. You can glue it onto your protein of interest, and now your shuttle protein is basically carrying a receptor for DNA on its back, goes out, picks up a DNA sequence, and then comes back inside. Lastly, people have been making aptamers, which are single strands of nucleic acids, either RNA or DNA, that bind very specific cell types. And fortunately, if you're lucky and your shuttle protein happens to have an aptamer that's already been developed to it, simply extend the sequence of your DNA to include the sequence of your aptamer. And that is another way by which you can get into a target organelle. So today I want to talk to you about how we can measure chemicals inside the lysosome. I'm going to talk to you about a sensor that measures chloride, which we call cleanser. And using cleanser, when we measured chloride, what we found was that, which was a real surprise, was that the level of chloride inside the lysosome was extremely high. In fact, we found that it is the organelle which has the highest level of chloride anywhere in the body. It's higher than even extracellular chloride. There's no other organelle that contains as much chloride as the lysosome does, which was a surprise to us because nobody had ever measured chloride in the lysosome before. But this gave rise to some new biological insight, where we found that the moment you perturb chloride and you make the chloride values lower in the lysosome, it affects the capacity of the lysosome to degrade its incoming cargo. So you can think of chloride to be sort of the digestive juices of the stomach, right? So the moment you have less chloride, you're not as efficient. You get indigestion. The cell gets indigestion. Okay. The next thing that I'm going to talk about is to build up complexity on this system, where now we say, okay, can we sense two ions at the same time in the same organelle? And so we built a device that can measure both pH as well as chloride. And using this, we were able to show that you can distinguish between cells that are derived from healthy individuals and cells that are derived from individuals with neurodegenerative diseases. There are some neurodegenerative diseases that are very difficult to subtype. I'm going to show you that using this technology, you can tell which type of neurological disorder they have, and you can also predict their responsivity to drugs. And finally, this being TIFR, fundamental research, I think the use of this technology for basic biology cannot be understated. I'm going to show you how we were able to measure a chemical called calcium inside lysosomes, which was a 30-year-old problem that could not be solved. which we now solved using this technology. And because we can now measure calcium inside these lysosomes, to show you that you can also use this for fundamental biology, where we've identified the first protein that brings in calcium into the lysosome. So here's a chloride sensing dye that people have known for more than 15 years. Again, if you take this molecule and you throw it on a cell, it goes everywhere, right? So there was a chloride sensor, except nobody could measure chloride in specific compartments with it. So what we did was we took this chloride sensor and made a DNA duplex with it, such that it had a 1:1 ratio of this green dye, which could sense chloride, and this red dye, which was a reference dye. And of course, the ratio of these two should give you a linear plot, and which from where you can just read out the concentration of chloride. And what we showed was you can get a very nice ratiometric sensor. So you can see the concentration, the fluorescence of this red dye doesn't change at all. But if you keep adding chloride, the fluorescence of this green dye goes off, right? And so we could use this. This was developed by Sonali Saha, who actually won the Zita Lobo Best Thesis Award before this work was published. And so I'm eternally grateful to TIFR for having taken a look at this thesis with unpublished work that described this chloride sensor and the use of it to measure chloride inside lysosomes, and decided to award this very coveted honor to her.
And
yes.
back to that,
yes.
Is it just a static quenching mechanism?
This is actually the reason why it's linear. So the question is, what is the mechanism of quenching? The mechanism of quenching is collisional quenching. Okay. And because it's collisional quenching, you will see that it is linear. Otherwise, if it was static quenching, it would be sigmoidal. And so then, yes?
So why is it sensing only chloride and not the other anions?
So actually, this particular dye also senses every halide. But inside the body, the concentration of other halides, all other halides other than chloride, is less than 0.1%. And therefore, inside the body, it's a chloride sensor. And so then what Sunena showed, Sunena was a joint student between Sandhya and me, she was a geneticist. So she showed that if you take these DNA devices and you inject them into nematodes, they go and label very specific cells called coelomocytes. And because coelomocytes are the putative immune cells of C. elegans, they express the scavenger receptor that takes these DNA devices and shuttles them into the lysosome. Okay? And so you'll see images like this. This is a single coelomocyte. All these little balls here are basically lysosomes in coelomocytes. And you will take an image in the red channel for the red dye, and an image in the green channel for the chloride dye. And we divide the image here by the image here to be able to get a value that's going to be color coded. So if it's a high value, then it's more red in color. If it's a low value, it's more blue in color. So literally, this is a heat map of chloride. So high chloride is red and low chloride is blue. And so what we were able to show was that this device performed as well inside a C. elegans, a live C. elegans, as it did inside a glass cuvette, which shows that this device is now performing an independent function. It's not being interfered with by the cell's own mechanisms of DNA control. It's an artificial device that's just sitting there and giving you a report of concentration. And so we said, okay, now that it's able to sense chloride, let us validate whether this is a chloride sensor or not. So we validated it in a lysosomal storage disorder model. I'll just tell you what a lysosomal storage disorder is. So as I said, a lysosome has about 50 different enzymes sitting inside it. And for every enzyme that goes bad due to a genetic mutation, it results in a different disorder. So if the enzyme that degrades mucolipids goes bad, and these kids have a mutation, then they will get a disease called mucolipidosis, where mucolipids cannot be degraded. So it just swells up. Then the cell dies, the tissue dies, the organ fails, and the child also unfortunately dies. So these are a bunch of rare diseases that are Mendelian disorders. They are genetic disorders where the child has just one gene mutated. And if it happens to be a gene in the lysosome, the child has a particular lysosomal storage disorder. Together, all of these lysosomal disorders number about 1 in 5,000, and that is in the Western world. Here, we don't even have knowledge of that such diseases exist, broadly. So this number could be higher. And so there's a disease called, yes?
What is the lifetime of your reporters in this? Because I presume there will be things in the cell that will attack your DNA. And
that's the next slide. But the lifetime of the devices can be measured. We've done that, and it turns out to be anywhere between 8 hours or 12 hours. That depends on the nature of the device. You've got to make this measure every time in a new system. But our measurements are made within an hour. So there's no problem at all. And that's probably a good thing because you're not perturbing your cell in any way, right? And it's a natural substrate, as you will see. And so we decided to look at, validate the sensor in a model called osteopetrosis, which is caused due to the misfunctioning of a chloride channel called CLH6 on the lysosome. So what we were able to show was that if you mutated proteins that were involved in chloride accumulation inside the lysosome, you could clearly see that chloride levels fell. But these two are proteins that are involved in the disease but not actually related to chloride levels here. And so we were able to validate this. You can see that the mutations here cause specific changes in chloride and no changes in pH. And so this was just to show that this sensor was indeed a pH-independent chloride sensor, right? And so the next thing was to ask, well, okay, in this particular disease model, we know that chloride is depleted, but what is the function of chloride in this? And so Kasturi, who's done a lot of the work that I'm going to talk to you about today, who's a Harper Fellow at UChicago, used an assay that had been developed by Sunena. What Sunena had tried to address was actually what Shudipta just asked, which is what is the lifetime of DNA devices inside C. elegans. So if you inject this DNA device inside C. elegans, it goes to the lysosome. The lysosome's job is to take up DNA that's floating from dead or dying cells and degrade it and then allow the body to reuse that material. And so this is a natural substrate anyway. So that means your DNA device is going to be degraded. So we measured the half-life by looking at the intensity of these spots and of fluorescence intensity, because once it's chewed up, the intensity goes out of the lysosome. And so we were able to show that it was about 8 hours. And so what Kasturi said was, well, I have just dysregulated chloride inside the lysosome. What happens to the lysosome's capacity to degrade DNA? So she's looking now at a longer time scale. First, the device comes in, you measure chloride, then you measure this look at the intensity over a longer time scale and ask how long does it take for the sensor to degrade. And what she found was that the more dysregulated chloride was, the longer was the half-life of the DNA device. So this basically meant that the less chloride you had, the lysosome's teeth became blunter and blunter. So then this motivated the next question, which was, okay, if I have high chloride, I have good degradation. I drop chloride, so now the cell has indigestion. I also know that there are a range of lysosomal disorders where the lysosome cannot chew up the material anymore. What happens to chloride in this condition? Right? So she took a number of diseases, C. elegans models for each of these lysosomal storage disorders, and found that chloride as well as pH were depleted. And they fell into 3 classes. One, those classes where only chloride was depleted, only chloride was dysregulated, but pH was normal. A bunch where pH was normal, but chloride was dysregulated. And a whole bunch of diseases where both pH was dysregulated as well as chloride was dysregulated. Why was this important? People had always known that when you have lysosomal disorders, pH inside lysosomes is dysregulated. But we found that chloride was also dysregulated, but to a much higher extent. This now gives us the sensitivity to look at, use this for a diagnostic, right? Because if I have much greater dysregulation, 10,000-fold higher dysregulation in chloride than I have of pH, then if I use chloride as a diagnostic, I may be able to diagnose these diseases earlier. And so, yes.
Just a question on,
Yes.
again going back to the first question you asked. I'm looking for the resolution on fluctuations.
Yes.
So I see steady state concentrations being
Yes.
reported here.
Yes.
Now, if I move from a normal cell to a disease cell,
Yes.
so if I can do that rapidly,
That's the next.
how rapid will
Yes.
be the sensor?
That's exactly what I'm going to address in the next part of the talk.
Okay.
Yeah. So, so I'm glad somebody's paying attention. And so, what we then decided to do was to say, okay, this is interesting. You can see this in worms, but worms are not humans. Sorry, Sandhya. And so what we did was we made what are called cell culture models of lysosomal storage disorders. So you can take fibroblasts, you can take skin biopsies from mice as well as humans. And what you can also do is you can inhibit specific enzymes using chemicals. And this is called a drug-induced model of a lysosomal storage disorder. You are artificially shutting off an enzyme and creating a lysosomal disorder in a cell. And you ask what happens to chloride and pH. And yes, we found that chloride and pH values did change in two different kinds of cell culture disorders, Niemann-Pick disease as well as Gaucher disease. And so this next part was done by Kaho, who is from Hong Kong, and I had asked him that I'm going to talk about your work, so please send me a picture of you that I can show to my colleagues in India. And he said, you have to show this entire picture of me. So I think he's trying to say hello and namaste to all of us. Anyway, so what Kaho decided to do, along with Kasturi, was to try and see whether we could leverage this capacity to sense, because both pH and chloride were dysregulated in some diseases, right? So we said, okay, let's try and see if in a single lysosome, we can measure both pH as well as chloride with single lysosome resolution. What does that mean? It means that if I can measure, take a single lysosome, I should be able to tell both the value of pH as well as the value of chloride for that lysosome. And so I'm just showing you the fact that you can take a pH sensor and stick it on to the chloride sensor. Just to tell you how the pH sensor works, if you make the solution a little acidic, this region is pH sensitive, it folds up and it changes fluorescence. So we now have three colors to look at. This green to red ratio, that's going to tell you chloride. And the orange to red ratio here, I'm going to call this donor to acceptor ratio here. This D to A ratio is going to give you pH. And so we were able to show that the pH sensing module is independent of the chloride sensing module. Regardless of whatever chloride value you have, your pH sensitivity of your device is retained. And the same is true for chloride. Regardless of whatever pH value you have, your chloride capacity, your chloride performance is the same. This is what I've just described to you. And so now you take this sensor and label lysosomes from skin biopsies derived from healthy individuals. This happens to be a fibroblast from Maulik in the lab, who there's no picture of him. I assume he's healthy. And so what we've done is, you see here, 600 data points for 600 lysosomes. So if you take any data point here, you can say, okay, this lysosome here has a pH value of so much and a chloride value of so much. And so you just look at the spread, right? It's very hard to see what this looks like, so we've converted it into a density plot. And so this density plot really shows you that your highest value, the most represented values are here, the least represented values are in blue. And what we're able to immediately see is that there are actually two populations. There are two maxima, one here and one here. And so we were quite intrigued and we said, okay, does this happen for everybody? Is it just like one? So there's this one high chloride population lysosomes here and low chloride populations here. Okay. And so when we looked at 3 healthy individuals, we found that all of them had this high chloride population. So then what we did was we said, okay, let's take skin biopsies from a repository where these people were suffering, had been diagnosed with different subtypes of a neurodegenerative disease called Niemann-Pick's disease. Niemann-Pick's disease comes in 3 flavors, A, B, and C. And so in all of these, you will see that this high chloride population is lost. And in fact, in many cases, the distribution becomes almost a monotonous distribution. It's not this 2 populations. And so it really meant that there was something, the stomach seems to be this high chloride population, right? And so we said, okay, let's try and see if we treat them with therapeutic, what happens. And so Niemann-Pick A and B result because there's an enzyme called SMPD1 or acid sphingomyelinase that has been mutated. And so it comes in 2 flavors. One is a very severe mutation where you have less than 5% activity, and a less severe mutation which has more than 10% activity. So the second one is called B, the severe mutation is called A. And the therapy for this is to actually give the person the enzyme. You eat the enzyme, right? This is called enzyme replacement therapy. And so when you treat the cell with the enzyme, recombinant enzyme, you see that this high chloride population has come back for Niemann-Pick A and Niemann-Pick B diseases. Niemann-Pick C is due to cholesterol transport. There is no therapeutic for this, okay? And many times these children are given, they think that these kids have Niemann-Pick A-B disease, and they actually have Niemann-Pick C. So they will give you recombinant acid sphingomyelinase, frequently misdiagnosed, frequently mistreated, right? And you will see that many of them won't respond. But there is one very bad therapeutic that is right now being put through clinical trials. It's ongoing at the NIH. And you have very little responsivity. So here you see 3 patients. This guy has responded somewhat. This guy, this is a non-responder. And this guy also has responded a little bit. Why is this important? Because if I am so unfortunate as to have Niemann-Pick A or B disease, or Niemann-Pick C disease, if I'm treated with recombinant acid sphingomyelinase, it takes about a year before I see anything, before I see any effect. And it costs $250,000 a year, right? That is the cost of the therapy. And so it would be great to know beforehand whether I need to be on this therapeutic or not. And so then the question is clearly that high chloride population is doing something. What is it doing, right? What is the role of chloride? So the lysosome is actually a bag of calcium. It's called an internal calcium store. And one of the ways by which it functions is to be able to release its calcium in a very targeted way, right? And this is one of the main functions of the lysosome. And so we thought, okay, what does it do in terms of the capacity of the lysosome to release calcium? And so what you do is, the lysosome is anyway a balloon. So what people basically do is they puncture the lysosome with a chemical that causes a particular channel to open, and then calcium just sort of rushes out. And you can look at calcium rushing out by putting fluorescent dyes in the cytosol of the cell. And you will see that for a normal lysosome, you have whoosh, a huge amount of calcium comes out. But when you deliberately reduce chloride and you then puncture this balloon, you find that there's very little calcium that comes out, right? So it affects the lysosome's capacity to release calcium, right? Note here that we could not actually see whether the lysosome's capacity to intake calcium was affected, right? And I will tell you why we couldn't do that. Technology doesn't exist. So what people do is that they look at how much calcium is released, right? If I want to know how much air is inside this balloon, I measure how much air this balloon can release. Yeah? Okay. So the second thing that we asked was, well, we also know that the lysosome's capacity to eat is compromised, but why, right? And so here's proof of that. So we've marked the lysosomes with two chemicals. One is a chemical called lysotracker, which goes and gets accumulated in the lysosome because the lysosome is a highly acidic environment. So if you try and puncture this lysosome, or you affect lysosomal integrity, what happens is that the lysosome will bleed protons. And so then what happens is the lysosome becomes less acidic. And when it becomes less acidic, lysotracker will not stain it. The fluorescence intensity of this red will go down. On the other hand, there's a chemical called dextran, which is a polymer that's going to be sitting inside, and it's fluorescent all the time. So even if the lysosome loses integrity, you'll still see that the lysosome is there. It's just not very, doesn't have very good integrity. So if your lysosome chews up a substrate, so you've labeled the lysosome with both these chemicals, because you have red and green there, it's going to look yellow. So that's what you'll see. And now you add a chemical called GPN. And so enzymes on the lysosomes will cut GPN, and what GPN does is it punctures the lysosome and causes holes in the lysosome membrane so that it loses integrity. And then what happens is lysotracker will leave, and now you will see only green if everything is okay with the lysosome. And what we showed was that if you reduce chloride, what happens is that the capacity of the enzymes that are present inside the lysosome to function are compromised. This happens to certain enzymes, and it's not across the board. Only some enzymes are chloride sensitive. And that was our basic finding. So chloride basically does two things. One, it's really important for lysosomal calcium release, which is important for the lysosome to fuse. And the second is, it's necessary for the activity of specific enzymes that are present inside the lysosome. And now, when we were doing this, yes?
So were the identities of these enzymes known beforehand, that they are chloride sensitive through biochemistry?
One of them was known. And the others we just scanned through to see, because we know that the assay works and we had a positive control. And so aryl sulfatase B was another enzyme that we found. Certain cathepsins, but not all cathepsins. Okay. And so when we were looking at this, we found that there was no way to measure calcium inside the lysosome, right? And then we said, well, why is this the case? This seems to be very fundamental. Are biologists actually happy with this, that they can't measure calcium inside the lysosome? And it turns out that they were very furious for the last 30 years and very frustrated that they couldn't measure calcium inside lysosomes. Many people told me that please solve this problem. So we sat and thought about it, and that's what I want to talk to you about. We've just managed to solve a 30-year-old problem. And I think quite a few lysosome biologists are quite excited by this. But I want to tell you what the problem is. Extracellular calcium is about millimolar. Inside the cell, in the cytosol, calcium is nanomolar. And this 10,000-fold difference in calcium is what drives all of biology. And the cell signals to itself by elevating calcium inside the cytosol to tell the cell to do a job. The master regulator that decides to do this is an organelle called the ER, which is really a bank of calcium, which is also millimolar concentration. And what it does is it opens channels that releases calcium, and this tells the cell, excites the cell, and sets off signaling. But again, this is not done by itself, right? I can't simply randomly, if the ER opens, then we'll all be dead. And so you have what are called internal stores of calcium. So these are acidic stores of calcium, little bags like the lysosome, the Golgi, and the mitochondria. All of them have small levels of calcium that they generally release small puffs of calcium that then causes calcium to be released. And so we've always wanted to look at these internal stores of calcium but could never do so. And so what I'm going to talk to you about is give you some examples of taking one organelle and one protein on one organelle. So whatever I'm saying, just multiply everything by 10. I'll give you 3 examples of how this happens. So for example, if you take this protein called TPC, the 2-pore channel, which is on the lysosome, which is one of the acidic stores of calcium, when its ligand binds, TPC will open, release a puff of calcium, and this causes the lysosome to, this causes the channels on the plasma membrane to open, which then changes the membrane potential, and this is really important for insulin secretion. In another flavor, just for any cargo to go down the endolysosomal pathway, you need to have normal trafficking that happens. So when early endosome and a late endosome or a lysosome and a late endosome dock, again, when the ligand binds to TPC, it releases a puff of calcium and these 2 compartments fuse. Lastly, the ER the lysosome can actually go close to the ER. When its ligand binds, it triggers a puff of calcium, and this causes the ER to then release its bank load of calcium. This is called calcium-induced calcium release, which is also extremely important. But none of these processes, you actually want to be able to sit inside the lysosome and watch the calcium change, which we've never been able to do. And why is that? Because every single calcium dye is pH sensitive, which means that you have this dye that lights up not only if there's calcium, but if the system is acidic, it's going to light up for that as well. And so if I take this dye and put it in the cytosol, yes, I can look at calcium, there's no problem. But if I put it in the lysosome, I don't know what I'm looking at, whether it's pH or whether it's calcium. And so what we said was, well, if you, and so whenever biologists used to look at acidic compartments and had to measure calcium, they'll run away from that problem. And so what we did was we turned the problem on its head. And we said, well, in this case, this dye is going to be sensitive. It's only sensitive to 2 things, right? So if we can measure the second thing, then we can introduce a correction factor. So if you take a duplex that's carrying this calcium sensitive dye and a calcium insensitive dye, this ratio is going to be sensitive to both pH as well as calcium. Now, if you attach onto this a pH sensor, such that in every lysosome, I know exactly what the pH value is. Then, if I know exactly how this dye's properties are going to change as a function of every single value of pH and calcium, then if I know the value of pH, I can introduce a pH correction factor and get the value of calcium. That's exactly what we did. So we showed that you can make this pH sensor, you can attach a calcium sensitive dye. This was synthesized by Nagarjun, who basically showed that you can get, you can see that how the calcium sensitive, the calcium sensing capacity, the KD, the affinity, changes as a function of pH, and you can describe that by an equation. And you can also see that using green and red we can tell calcium, using orange and red you can tell pH and calcium. And so I'm going to add a second layer onto this. So if you take your coelomocyte, which is now labeled with your device, you will take 4 images: the donor channel, the acceptor channel, the orange channel, and the red channel. This, I already told you how to get a pH map. This is going to be pH and calcium. And so what we were able to show using this was that you could measure the performance of the device, not just inside a cuvette. You could take it at every condition of pH and calcium inside a worm. And you can plot it. And this was done by a very talented graduate student, Anand, who was from IIT Guwahati. He's just finished his BTech and he's now a fourth year grad student. And this was all done by Anand. So I showed you how to get this D by A map, right? We already know you can describe this D by A as a function of pH by an equation. You can substitute the value of using this at every single, you can convert this D by A map into a pH map, right? We already know how pH changes as a function of the dye binding affinity. I showed you that map earlier. And so you can convert this pH map into a KD map, right? And now I have the O by R map, which is pH and calcium, right? And so if you multiply these two, using this equation that describes exactly how this changes, you can basically get exclusively the calcium map. I'm taking a little bit of time to tell you this because these are the first acidic calcium maps in history. And the reason I want to take a little bit of time is to tell you that this pH sensitive, pH sensing dyes have been known for about 50 years. Calcium sensitive dyes have been known for 40 years. The concept of endocytic tracers have been known for even longer than that, 40, 45 years. But we could not put all of these things together, right? It took a simple DNA duplex, which can act like an endocytic tracer, and take each of these components, put them on a 1:1 ratio to make a precise device that allows you to make a measure and be able to get a value of calcium. And so then what we were able to show was we could use this device and look at the calcium values in each as a function of endosomal maturation. So what we found was, of course, pH became more and more acidic as the system was undergoing endosomal maturation. This is nothing new, we know this. But this is new. Calcium values are fairly low in the early endosome and late endosome. Suddenly in the lysosome, there's a 35-fold increase in calcium. And so immediately we asked, who is bringing in calcium, right? And it turns out that we went and said, okay, who are all the lysosomal calcium importers that were known? We searched for weeks, we found nothing. We found large numbers of proteins that released calcium, right? So we know people who are releasing calcium. Somebody is putting calcium inside the lysosome. Who is that? And so we then found that there was one report back in 1985 which said that we must have lysosomal calcium importers, but no genes were mentioned, no candidates were mentioned. So we know nothing about lysosomal calcium importers other than the fact that we definitely have them. Okay, and so what we did was we just asked, well, we know who is the best calcium importer that's present on another organelle called the endoplasmic reticulum. And we know the lysosomal proteome. We know every single protein on the lysosome. This is public knowledge. So we went to the public database and we said, which of these proteins looks exactly like this importer called SERCA, right? And we found 8 candidates. And I really liked this guy, ATP13A2, because it looked so similar. So we said, okay, let's look at whether ATP13A2 is going to function as the importer. And so here's the system. The system we are going to look at is a certain protein called CUP5, the human homologous TRPML1. This is a very well-known calcium releaser. If it opens, calcium rushes out. Very, very well defined. And so what I'm going to show you are a series of experiments that will say that this protein CATP6, in C. elegans this is called CATP6, this protein CATP6 performs the exact opposite function of CUP5. So if CUP5 is releasing, this guy is going to be pulling in calcium. And how we're going to do it is the following. You're going to use this in the worm, and you're going to see 3 different phenotypes, or 3 different properties, physical properties. One is, if you take, your worm is either going to be dead or alive. So if CUP5, this gene is mutated in the worm, both alleles, that worm is dead. If you somehow compromise the function of CUP5 in the worm, you're going to see a second phenotype, a second physical property. That is, this is going to be a subcellular phenotype. Your lysosomes are either going to be a normal size, or because you've created a lysosomal defect, you can't degrade cargo, boom, it just becomes like a balloon. And the third is the value of calcium. If you block a calcium releaser, so that's like a pressure cooker that cannot release calcium, right? If you plug it, then the value of calcium is going to become very high. And so what we were able to show was the following, that so CUP5 is a leaker, right? So if you delete it, the worm is dead, you have huge lysosomes, and you have high calcium. If you delete the accumulator as well in the background of this, it's going to set right this. Instead of having dead worms, you will have live worms. These worms will have normal sized lysosomes, and the calcium values are going to come back to normal. And so what we showed was that your worms were alive. We showed that instead of huge lysosomes now, the lysosomes came back to a normal size. And we were also able to show that if you delete a leaker, calcium values increase. If you delete an accumulator, which is the SCAT-P6, calcium values plummet. If you delete both, calcium values come back to normal. And so at 3 levels, this gene is doing the exact opposite function of CUP5, and that is it's facilitating import. And so now the question is, what is this ATP13A2? It's also called PARK9, and it happens to be one of the major risk genes for Parkinson's disease. And so what we did was to ask, does it actually do this job in humans? And so we took normal humans' lysosomes and measured calcium. We got a certain value, 300 or 400 micromolar. And then we took skin biopsies from Kufor-Rakib syndrome patients. Kufor-Rakib syndrome is it's very sad. These patients are male patients who have very severe form of Parkinson's disease. It's early onset. They're just 35 years old or something. They basically have a single mutation, and the single mutation results in the ATP13A2 not being able to leave the ER. So it's stuck in the endoplasmic reticulum. As a result, the body is, the cells are making ATP13A2, they're just not on the lysosome, right? So there's, that's why these patients are ill. And if you measure calcium there, it's completely, it's down 12-fold, which basically means that ATP13A2's job is to bring calcium into the lysosome. People have been wanting to understand what this protein does. There are meetings that are held on this protein, but it was quite interesting for us to find that it has this function of importing lysosomal calcium.
And it's like some sort of an active transport?
It is an ATPase. It's a calcium ATPase, right? It's a P-type ATPase. And so the reason why is it we who are chemists have found out the first lysosomal calcium importer? And the reason is the following, right? That if you want to look at a leaker, I put my calcium dye outside a neutral environment, so I can pop this balloon and see how much calcium comes out. So it's easy to investigate leakers. But if I want to look at importers, my probe has to be inside, right? And I can't do that. So now, since we can do that, now we can start looking at leakers. And so we want to make this probe widely available to all people who are studying lysosomal calcium releasers, because I don't know if you noticed, but for every lysosomal calcium releaser that's known, mutations in these causes a different neurodegenerative disease. And so it's highly unlikely that you would have so many releasers and just one importer. So now what our lab is doing is to try and see if we can identify new importers. But we want to make the probe widely available to everyone. And so with this, I'm going to just close and say that now with the capacity to measure chemicals inside live cells, one can actually start looking at the responsivity of cells to drugs, which I talked to you about. But also you can use it for discovery biology, which is, I think, important. Fundamental research, I think, drives all application. And I just spoke to you about the capacity of this technology to be used for personalized medicine, or rather precision medicine. We didn't say it, some editorial said it, and so that's why I put it here. And very recently, which I've not been able to talk to you about, is that we've now been able to look at, measure enzyme cleavage. All these ions at the end of the day, ion fluctuations result in enzymes not being able to act. And so now we have the capacity to measure enzymatic cleavage inside these organelles. This I didn't have time to talk about. It's just come out this month in Nature Nanotech. But I want to end by quoting Lord Kelvin, who says, when you can measure what you're speaking about and express it in numbers, you know something about it. When you cannot express it in numbers, your knowledge is of a meager and unsatisfactory kind. It may be the beginning of knowledge, but you have scarcely, in your thoughts, advanced to a stage of science. And so with that, I just want to say, I want to thank my wonderful group. Actually, all of this work was started by one very brave PhD student who is now a postdoc sitting in this audience somewhere here. His name is Shovik Modi. He's now working with Sandhya. I'm sure he shrunk inside his chair and I cannot see where he is. But it was a time when people were thinking that we were crazy to take DNA and measure ions inside cells. But I don't know where you are, Shovik, but I hope you can see what your work has grown into. And so all of this work was started by, I've acknowledged most people along the way, but the lab really is a mix of people from all areas. We have a chemical engineer, photophysicist, photochemist, a polymer chemist, biochemist, biotechnologist, geneticist. We collaborate heavily. For this particular work, we collaborated with Dimitri Cranch, who gave us Parkinson's patient cell line. But most of the work was actually done in-house. And I thank these organizations for their funding, TIFR for believing in my vision and hiring me 12 years ago at NCBS, and I'm happy to take any questions. Yes.
So for your different sensing strategies,
yes.
Using these aptamer-based devices.
Yes.
So I assume for different strategies, the aptamer designs are different, like the nucleic acid.
Yes. Yes.
So my question is, how do you select the different types of different aptamers for different strategies? So which
yes. So I've been asked to repeat the question. So the question is, if you want to sense different analytes, how do you decide which aptamer to use? So what I mentioned, what I show you here, each sensor takes the rate determining step is actually sensor development, which we take for granted. And so it's really by trial and error. So either you can develop an aptamer by yourself, which is the long and painful way to do it, or you can go to the literature and you can see aptamers that already exist. You can do an affinity maturation and increase the tightness of binding of that aptamer, or if it's too tight, you can make single nucleotide mutations and change the KD of the aptamer. And then you do in vitro measurements to understand whether it has the sensitivity or the required KD for you before you make a sensor. Yes.
It was, of course, a fascinating talk.
Thank you.
I have two related questions. One is, what about some kind of calibration? I mean, do you have some way to find out that what the actual concentration that you're measuring is really very close to the
yes.
concentration?
Yes.
And secondly, how sort of sensitive are you to the environment, in the sense that are there some ions which if present, will make your sensitivity less?
Can they interfere? Yes. So the first question is, do you do any calibration, or are you pulling these numbers out of thin air? And the second, so which I will answer first. So actually what I didn't show you is very rigorous calibration procedures, which are not just done in vitro, they're also done in vivo. I don't know if I have a slide for calibration. One second, give me a minute. Yeah, okay. Oh, okay, it's doing that. One second. Yes, so this is a very data heavy slide, which is why I skipped it. But it's just to tell you that you can look at the device, you can assess device performance quantitatively if it's a binding based sensor from these two parameters. One is the fold change in signal, and the other is the half point, the point at which you get half the response. You can do this in vitro, and then what you also do is you perform a similar experiment in the worm. You take the worm and you can clamp the ion concentration in the worm using what's called a clamping buffer, which sets the value of ions inside to a known value of the buffer you've added outside. And then you take a look at the sensor's response. And then you can compare. In vitro is in pink and in vivo is in gray. And so then you can actually quantitatively compare the performance of your thing. And now your sensor is actually sitting in vivo. So whatever is going to interfere should have interfered. But if it's giving you the exact same performance profile, then you know that it's sensitive to your ion of interest. And the second question was, I forgot.
How sensitive is it then?
I answered that, yeah.
And the other was calibration you have already also.
Yes, yeah. Yes. Yes, Shamun.
Yeah, you can score.
It's already answered. Yeah, I answered both his questions.
No, because, sorry.
I have a question for you asking.
Yeah, because Roop has.
Oh.
Go ahead. No, so most of the thing is the chloride that we've been talking
Yes,
about, and the calcium.
Yes.
And the
yes.
So what are the counter ions for the calcium and the chloride? When you down-regulate or up-regulate chloride,
Yes.
what is the counter ion? Most of the calcium perhaps exists as the calcium chloride.
So in fact, people used to think of chloride as nothing but a counterbalance for protons or for calcium. So that is what sets the membrane potential inside this bag. You have chloride concentration on the outside, which is different from chloride concentration inside. Same for calcium, for potassium, for protons, and that sets your membrane potential. So as you know, yes?
When you have huge 6 orders of magnitude difference between the outside and inside,
Yes.
is that, wouldn't that lead to too high membrane potentials? What is the magnitude?
So on the plasma membrane, so in fact I was going to, the talk that I would have given would have directly addressed membrane potential, because we've just been able to make a membrane potential sensor and we've been able to measure membrane potential at the plasma membrane, which people have measured already, but we've also been able to measure the membrane potential of every single organelle inside the cell. And people didn't know what these membrane potentials were. Turns out that the lysosome has a membrane potential of plus 110 millivolts. The plasma membrane has a potential of minus 70 or minus 50 millivolts, depending on the cell line. But many of these organelles have membrane potentials of anywhere between plus 130 to plus 100 plus 130, plus 140 millivolts.
Negative inside?
Yes. So here's the interesting thing. It's actually supposed to be negative, but the convention for organelles is reversed. So they change it to plus because that's how they insert their electrodes. Yeah. So that's the.
This kind of a continuation
Yes.
of that, could some of your calcium importers,
Yes.
if you want to find
Yes.
be using the proton gradient actually?
Yes. So here's the thing. we are planning to do this screen for C. elegans. So if there are C. elegans geneticists here who want to make a big splash, contact me later. But we have a capacity to identify now every single gene that will lead to lysosomal calcium accumulation. Some of these could be ATPases. In fact, ATP13A2 is a calcium ATPase. Some of them could be associating with a protein that is bringing in calcium. Like for example, CLH6 has this accessory factor OSTM1. So if it doesn't have the accessory factor, it's not going to work. And finally, some of these could actually be taking the organelle to the plasma membrane and then activating a crack channel to then achieve calcium uptake. So there are any number of mechanisms that one can think about.
Ankur.
So you beautifully see this pH and this chloride, right? Do you see a third pair like HOCl crop up now?
Yeah, so we have, what I didn't talk about was our recent paper in Nature Chemical Biology that's ASAP, it's on ASAP, where we've shown that we can measure HOCl, which is the first example of measuring a reactive species inside an organelle called the phagosome. And in fact, a direct measure of HOCl can be made. And what we find is there's a very slow buildup of HOCl once you have the phagosome. But the moment the lysosome fuses, there's a huge jump, there's a burst of HOCl that comes about. And what we think is the following, that myeloperoxidase already may be present, right? But then it's also the delivery of a huge amount of chloride into the phagosome upon the fusion of the lysosome. That gives you the substrate chloride for myeloperoxidase to make HOCl. Myeloperoxidase, you can have some amount of myeloperoxidase that might be present. But actually when the lysosome fuses, it will deliver both myeloperoxidase as well as chloride, right? But everything else that the system needs is already there in the phagosome. So now you have fire and oxygen and yeah.
Yes.
Beautiful talk and excellent work that you have brought into this particular field. So I would like to know how much far are we away from actually monitoring different ions in both space and in time across the entire micron length cellular envelope. So, so you have shown that this can be done in micromolar to millimolar concentrations.
Yes.
Can you go down to nanomolars and all the way up to millimolars in time? So how far are we away with these kinds of tools?
So what is the kind of change that you will, what is the resolution that we are able to see? I think that is going to be determined by microscopy, right? Because I think chemicals are going to set, will set the threshold of where you can image and what quantity you can look at. After that, when you're looking at resolution, it is going to be dependent on microscopy and optics. Yeah.
Okay.
Yeah.
Okay. I think there are more questions. Of course, you can always interact with the speaker at the table. But we want to leave the speaker representing this nice.
Thank you.